Welcome to Lesson 3 of Bioenergetics Photosynthesis. As always, just a few tips on how to get the most out of this lesson. And here is the run through of the lesson. Same structure as always, and you'll see the resources that you'll need for the retrieval, the whiteboards or pen and paper, and revision cards, and of course there will be resources for you to print out from Show My Homework. So if you haven't already, can you please pause and organize? Okay, so as always, we're going to start today with some retrieval practice, and we'll start with biology. So if you could please pause and retrieve. Here are the answers to the biology retrieval. Can you check your work and make revision cards? So pause and revise. Chemistry retrieval, pause and retrieve. Here are the answers to the chemistry retrieval. Can you please mark yours, make revision cards if you need to, and pause and revise. Here are the retrieval questions for physics. Pause and retrieve. Here are the answers to the physics retrieval. Mark your work, make revision cards. So can you now pause and revise? Here are the answers to yesterday's practice. These are the first 5 questions and their answers. So if you could please pause and check. Here are the next 2 answers to the questions you were given yesterday. Please pause and check. And here are the last 2 questions. So please pause and check your answers. Before we move on to teacher instruction, I just want to recap over yesterday's lesson. We worked on the required practical, which was to investigate the effect of light intensity on the photosynthesis of pondweed, and we wrote a method which you might want to look at again now, and we looked at possible errors in the method for that investigation. Today's teacher instruction. For this, you will either need to print out the resources from Show My Homework or use the slides that are shown in this video. So these are the resources you will need. You'll need this sheet, which gives us some data. You will need graph paper because we're going to plot a graph using that data. And you will need this table that we're going to fill out as I talk through limiting factors. So if you haven't already, can you pause and organize? Here is the first sheet, which has got the table of results. And what we're going to do, we're going to use this table to plot a graph. So we're going to analyze these results. But the graph we're going to plot is going to be the distance the light source is away from the pondweed against the mean number of bubbles of gas produced per minute. So the first thing we've got to do is work out the means for each of the distances. So the way we do this, if you remember, to calculate a mean, we add up all the values for that particular distance and divide by the number of values. Well, as you can see, we've got 3 values. For the 10 centimeter distance, we've got 46 bubbles per minute, 49 bubbles per minute, and 49 bubbles per minute. So we need to work out the mean. So we add these 3 numbers together, which comes to 144, and then we divide by 3 because there are 3 sets of data that we're using. And 144 divided by 3 is 48. So if we write that in, at 20 centimeter distances, we have 10, 11, 15. If we add those together, we get 36. 36 divided by 3 is 12. Okay. For the 30 centimetre distance, we've got 19. 5 plus 6 plus 8 is 19. Divided by 3 is 6.3 recurring. But we can't have a greater resolution than the data we've collected. So we've got to round that number. So 6.3 recurring becomes 6. We've then got 2 plus 3 plus 4 is 9. Divided by 3 is 3. And then finally, we've got 5 divided by 3 is 1.67. Again, we can't have a greater resolution than our data, so I'll round that up to 2. This mean data is the data that we are going to use to plot our graph. Okay, so what we need to do now is to get the graph paper. Hopefully you've got the graph paper. Here it is under the visualizer, and I'm now going to draw the graph with you. So you'll need a ruler, a pencil, and a rubber if you make a mistake. Now I'm going to be drawing in pen so you can see it. When we come to the drawing of the line of best fit, I'll change to a pencil just in case I make a mistake. So first things, we need to draw on the axes. So at the bottom is going to be distance. Now we've got 5 distances, so I'm going to go 5 large squares across. There's 1, 2, 3, 4, 5. Okay, so if I label this, so distance of 0, 10, 20, 30, 40, and 50. So this is our x-axis, and we need to label the axis. So it's the distance between pondweed and light source. So it's distance between pondweed and light source. And that distance is in centimeters. Remember, we always put on the units. So that's my x-axis. Hopefully you can all see that okay. And then up the side, if we look at our means, our largest mean is 48 bubbles. So that takes me all the way up to the top of the graph. Okay, so if I now place in my numbers, so 10, 20, 30, 40, 50, okie dokes. And that is the mean rate of photosynthesis. So it is the mean rate of photosynthesis and basically that's the bubbles of gas per minute. Okay, so there are axes. Okay, so hopefully you can see those and you've drawn those yourself. Now we need to plot the results. So if we look at 10 centimeters, we have 48 bubbles. So 10 centimeters, 40, 1, 2, 3, 4, 5, 6, 7, 8. So we have 48 bubbles being counted in a minute when the lamp is 10 centimeters away from the pondweed. At 20 centimeters, we've got 12, so that's 10, 11, 12. At 30 centimeters, we have 6, So that's 5, 6. At 40 centimeters, we have 3, 3. And at 50 centimeters, there are 2 bubbles per minute. 2. So there is our data plotted. Now, obviously, looking at that, it is a curved line. So what I'm going to do now is draw in the line of best fit, but I'm going to use a pencil just in case I make a mistake. With my pencil then, I'm going to draw the curved line of best fit. There. So that's our line of best fit. Okay, it's obviously a curve, not a straight line. So it's non-linear. Right, now that we've got our line of best fit, we can now analyze that and come to some conclusions. So that's what we're going to do now. So we're going to write our conclusions onto the graph. The first thing to note is how light intensity is related to distance, because remember, we're looking at the effect of light intensity on the rate of photosynthesis. And as we increase the distance between the pondweed and the light source, the light intensity becomes less. Okay, it decreases. So if we just make a note of that, because we're going to be talking about distances and light intensity when we write down our conclusion. Note. Distance between pondweed and light source are related to light intensity. Okay. As distance increases, light intensity decreases. Okay, so that's just a little note so we can remember the relationship between distance and light intensity. What the graph shows is, if we look at this curve, it shows that as light intensity decreases, i.e. the light source gets further away from the pondweed, the rate of photosynthesis also decreases, and that's shown by the number of bubbles per minute decreasing with increasing distance. Okay, so let's make a note of that. As light intensity decreases, the rate of photosynthesis decreases. Shown by the number of bubbles per minute decreasing with increasing distance. Right, let's look at some examples then from the graph. So just to prove what we're saying. So for example, at a distance of 10 centimeters, the rate of photosynthesis is 48 bubbles per minute. So let's write that. At 10 centimeters, the rate is 48 bubbles per minute. And if we look at 20 centimeters, if we double the distance, the rate is just 12 bubbles per minute. So at 20 centimeters, the rate of photosynthesis is 12 bubbles per minute. So that shows us that as we're increasing the distance, we're reducing the light intensity and the rate of photosynthesis is decreasing. Because of course, plants need light, they specifically need light energy to photosynthesize. Those of you doing the higher tier, you need to know about the inverse square law. So I'm just going to talk about that now and what that means. So this is for the higher tier students. Okay. Now, if we look, when we double the distance, the number of bubbles goes down fourfold. Okay. Now, there's a reason for this, and that is because when we double the distance, the light intensity decreases by a factor of 4. So let's just write that down first of all. By doubling the distance, the light intensity decreases by a factor of 4. Okay, and this is known as the inverse square law. Okay, so it's known as the inverse square law. And in fact, there's an equation for working out light intensity related to distance. And it is light intensity is proportional to 1 over the distance squared. Okay, so we just square the distance. Right, so what does that tell us about what we've seen in our results? So we can say, therefore, the rate should decrease by a factor of 4 as 4 times less energy when the distance is doubled. Okay. Now that is shown in this example up here. So we started at 10 centimeters. We had 48 bubbles per minute produced. We double the distance to 20 centimeters. The number of bubbles produced has gone down by 4. Okay, 4 12s are 48, or 48 divided by 12 by 4 is 12. Okay, so we can see that in this example. So we can say this is shown in the example above. This is shown in the example above. So does it work when we go from 20 centimeters to 40 centimeters? So we double 20 to get 40. So let's have a look. So also at 20 centimeters, the rate is 12 bubbles per minute. And at 40 centimeters, the rate is, so 40, 1, 2, 3 bubbles per minute. Okay, so we can see, can't we, that I've doubled the distance that the light source is away from the pondweed. And if we look at the results, the rate has decreased by a factor of 4. This evidence backs up the inverse square law, but because we can see that if we are doubling the distance, the rate of photosynthesis is reducing fourfold, because the light intensity, the amount of energy, is being reduced fourfold. Let's now get back to our original sheet and look at the question on limiting factors. The question we're looking at is, what are the 3 limiting factors of photosynthesis? So first of all, what is a limiting factor? And a limiting factor is simply any factor which, when it is in short supply, limits or reduces the rate of photosynthesis. So let's write down that definition first of all. So it is a factor which, if in short supply, limits or reduces the rate of photosynthesis. Now, there are 3 limiting factors that we look at on our specification, and they are light intensity, carbon dioxide concentration, and temperature. What we're going to do now is look at these 3 limiting factors in more detail. I mean, there are other limiting factors such as amount of chlorophyll and of course the amount of water, but on our syllabus these are the 3 that you'll be asked about. So if you can get the next sheet, and we'll look in more detail at these limiting factors. The first limiting factor we will look at is light intensity. So if we write light intensity in the first box. Okay, now we need a sketch graph to see what happens as we increase light intensity. So I'm just going to sketch this graph here. So I'm going to do the axes. There we go. So we've got light intensity on the x-axis. And we've got rate of photosynthesis on the y-axis. Okay, so what does the graph look like? Well, as the light intensity increases, the rate of photosynthesis increases. Okay, and then we get to a point where it levels off. meaning if we increase the light intensity, the rate of photosynthesis no longer increases. So I'm going to talk about these as two sections. So this can be section 1, and where it levels off, section 2. Okay, so describe what is happening in the graph. So first thing we can see, when light intensity is 0, rate of photosynthesis is 0. So when light intensity is 0, the rate of photosynthesis is 0. So that's the first thing we can see. We can also see in section 1, as the light intensity increases, the rate of photosynthesis increases. So in section 1, which is here, as light intensity increases, the rate of photosynthesis increases. Okay, so that's what's happening in section 1. In section 2, we can see that as the light intensity continues to increase, the rate of photosynthesis actually stays the same. It levels off. So in section 2, as light intensity increases, the rate of photosynthesis stays the same. As I've said, it's leveled off, or it levels off. Okay, so that's what we can see happening in the sketch graph. Now we need to explain why it's happening. In section 1, more light provides more energy, which increases the rate of photosynthesis. So in section 1, more light provides more energy, which increases the rate. Okay, so what does that tell us? It tells us that in section 1, light intensity is the limiting factor. So light intensity is the limiting factor. This means that the rate is not as fast as it could have been because there's not enough light energy. In section 2, the rate no longer increases, and another factor is doing the limiting, and that's either going to be temperature or carbon dioxide concentration. So let's write that down. In section 2, the rate no longer increases, and another factor is limiting. Okay, and that's either temperature or carbon dioxide concentration. Okay, so that is light intensity covered. So we've got the sketch graph, we've got a description, and we've explained why it happens. Now let's look at carbon dioxide concentration. So there's our heading. Now the graph for this looks almost identical to the graph for light intensity, except our x-axis is carbon dioxide concentration, not light intensity. So if we just draw a sketch graph, so we've got rate of photosynthesis, again on the y-axis, but this time we've got carbon dioxide concentration on the x-axis. Okay, so there are two axes, and again, starting from 0, as the carbon dioxide concentration increases, the rate of photosynthesis increases until we get to a point where it levels off. So again, I'm going to talk about section 1, which is here, and section 2, which is where it's leveled off. There is our sketch graph. So let's describe what it shows. And again, it's going to be along the lines of light intensity, but for carbon dioxide concentration. So when carbon dioxide concentration is 0, the rate of photosynthesis is 0. I'm just going to use rate now. I'm just going to write rate, but I mean rate of photosynthesis, so remember that. Let's have a look at the different sections then. So in section 1, we can see that as the concentration of carbon dioxide increases, the rate of photosynthesis increases. So we'll write that down in section 1. As carbon dioxide concentration increases, the rate of photosynthesis increases. Okay, and then in section 2, as the carbon dioxide concentration increases, so here's it's increasing, the rate of photosynthesis stays the same or levels off. So in section 2, as carbon dioxide concentration increases, the rate of photosynthesis stays the same. It's leveled off, so it levels off. Okay, so that's a description of what we can see in the graph. Let's look at the explanation. What do we know about carbon dioxide? Well, we know that carbon dioxide is a raw material for photosynthesis. Okay, so we can write that down first of all. So carbon dioxide is a raw material. It's one of the reactants for photosynthesis. Okay, So if we increase the concentration of carbon dioxide, that means we've got more carbon dioxide. And if we've got more carbon dioxide, that means there are more collisions that take place between the carbon dioxide and the water, thereby increasing the rate of photosynthesis. Increasing the carbon dioxide concentration means more raw material, more collisions with water, which increases rate. So that's just the background information. Now looking at the graph, we can see in section 1, carbon dioxide is the limiting factor. So in section 1, carbon dioxide concentration is the limiting factor. Okay, so we can see that because if we increase the amount of carbon dioxide, the rate increases. We know this because rate is not as fast as it could have been as there's not enough carbon dioxide present. In section 2, the rate no longer increases as another factor is limiting. So in section 2, rate no longer increases as another factor is limiting. And this time it's either light intensity or temperature. Okay, so that is light intensity and carbon dioxide concentration covered. The third factor we need to look at is temperature. So we'll just address the box. And we'll draw the sketch graph. Now this sketch graph is slightly different. So we'll draw the axes. As always, on the y-axis we've got rate of photosynthesis. And on the x-axis, this time we've got temperature. Now the graph starts off the same. As temperature increases, the rate of photosynthesis increases until we get to a point where increasing the temperature causes the rate to decrease and eventually photosynthesis stops. Okay, so let's describe what's happening. Again, I'll split it up into sections. So if we've got section 1, the increase, and section 2 will be the decrease. Okay, right, let's write what we can see. So simply we can see, first of all, in section 1, as the temperature increases, the rate of photosynthesis increases. So in section 1, as temperature increases, the rate of photosynthesis increases, So that's the first thing we notice. Then, as the temperature continues to increase, the rate decreases and eventually stops. So in section 2, as temperature increases, the rate of photosynthesis decreases and eventually stops. The explanation then. So if we look at section 1, increasing the temperature causes more collisions between the reactants. Okay, so let's write that down first of all. So in section 1, increasing the temperature causes more collisions between the reactants. This increases the rate of photosynthesis. Also, the enzymes involved in photosynthesis, they work faster because of the increased temperature. So also, the enzymes involved work faster. Okay, so that's why as we increase temperature in section 1, the rate of photosynthesis increases. But in section 2, as we increase the temperature, the rate decreases. Okay, so we need to explain why that happens. So in section 2, increasing temperature, or increasing the temperature, causes a decrease in photosynthesis, or in the rate of photosynthesis. As the enzymes involved are denatured. Okay. If you remember, denaturing is where the active site of the enzyme changes shape, so the substrate no longer fits. So they are the 3 limiting factors that we need to know about: light intensity, carbon dioxide concentration, and temperature. What I would like you to do now is embed all that information that you have gleaned today. So this is where you'll need your mini whiteboards or your pen and paper or someone to quiz you on the 3 limiting factors. So if you can pause and embed. Now that you've paused and embedded, it's time to do some practice. Again, the resources are on Show My Homework, but I've also got them here. So this is the first 4 questions that I'd like you to do, and these are the second 4 questions I would like you to do. So what I'd like you to do now then is pause and practice. Do some slop. We will discuss the answers to the practice questions in lesson 4. Next lesson, we'll be looking at limiting factors of photosynthesis graphs involving 2 or 3 factors. We'll be doing more slop, and we'll be doing ultimate retrieval. Until the next lesson, do some retrieval practice, complete the slop, share the video, and remember at the end of the week you'll be getting a recall test on the work you've done. See you soon.